So we mentioned earlier the three different types of canal systems that are used, and one of them was asconoid. And we said the asconoid, the collar cells are located in that central spongocyl, in that central cavity inside of a sponge. And so here's the osculum. This is where water would flow in through a channel, and then into the spongocyl, and then out of the osculum at the top. And inside the spongocyl, you have all these different collared cells lining the inside. That, by definition, would make this an asconoid type of sponge. We also have synchronoid. Synchronoid, they have these channels that lead towards the spongocyl in the center. And in these channels, we have all of the collar cells, right? So the water flows in to the osculum and into the little channel, and the food particles and nutrients will be filtered out by the choanocytes in these little channels, and that is a synchronoid. And the last one was the Lucanoids. The Lucanoid has a very complex system. There's just lots of long channels, and instead of one big spongocyl, like this is the spongocyl here on this one, instead of one big one, they just have several small chambers. And in these small chambers, this is where you're going to find all of the collar cells. So the canals run into small chambers and then out, and if the collar cells are in these small chambers, then this is a Lucanoid. So again, if it's one big hole in the middle, spongocyl in the middle, then you have collar cells lining that, then it is an asconoid. Synchronoid have their choanocytes or their collared cells are in the channels themselves. And then the Lucanoids, the collared cells, are in the chambers, which are linked together by all the different canals. So I hope that helps you understand a little bit about the differences between the main types of canal systems that are used. It's just where are the collared cells located. So let's look into a little bit more of the other parts and the other types of cells that can be found within sponges. So we have the mesohyl, the spongocyl, the spicules are some of the body parts. So the mesohyl is just the area in between. So you have the outside of a sponge and you have the inside of the sponge, and then you have that the big canal in the middle, the spongocyl in the middle, right? But what's in between the outer layer of cells and the inner layer of cells? And that's going to be a lot of mesohyl. It's just an area that's in there. There's different things in there, but it's just the middle of the sponge's body. Then you have the spongocyl, which again we say it's the large outcurrent cavity, which is centralized in a lot of them, and it's where the water goes in. It's kind of like a chimney stack that raises the water up. As the water comes in from the sides, it then goes up and out the chimney through the middle. And then again, that's the spongocyl. And then you have the spicules, which you mentioned earlier. These are hard structures. These are often they're needle-like, but they're used for structural support, skeletal support inside of the sponge. And they can be made of three different things. They can be made of silica, calcium, or spongin. And what they're using to make their skeleton out of is often things we can use to help classify them, because if these all use silica, they're probably related. If these all use calcium, they're probably related. And same thing if they all use spongin, then they probably share a common ancestor. But those are some of the things that are found on the inside. But we also have some of the different cell types. So we have the pianocytes, which are again the flat cells that are on the outside. They lock together, make like a surface, like a covering surface for the external part of the sponge. The cells are so flat; they do have a nucleus, but the cells are so flat that the nucleus is often found deeper into the sponge, and whereas most of the area of the cells on the outside, the nucleus actually be deeper down on the inside. If you looked at it from a side view, it would look like a letter T with a teardrop at the bottom, and that teardrop has the nucleus, whereas the flat part, the top part of the T, is the actual cell that covers the outside. They have the porocytes. These are the cells that can act as a channel. They actually have a hole that runs through them. So imagine a cell that acts as a channel. The entire cell is like a cone. There's a hole that runs through it. And there's an inside and an outside based on the water direction. So the water flows in through the forciple and out of the apiple. sides of the porocytes. So two words to throw at you there. So the procypal, which goes in, and the apical, which is where it comes out of the cell. And again, the porocytes are cells that act as channels. You do have channels that run through sponges, but not all channels are porocytes. Some channels are just that, they're just channels that run through. You have the choanocytes, which we talked earlier. These are the collared cells. These are the easiest ones to identify. They're cells that are collared and they have a flagella. The beating of the flagella is what causes the water to flow and causes the current that sucks water in through the sides and out through the osculum. And then there's a collar that's right at the base of the flagella that acts as a net that filters out and scoops up the particles floating through the water. The arachnocytes are also called amoebocytes. You'll hear me call them amoebocytes more often. But they're amoeba-like cells that actually move through the mesohyl, and they do a variety of functions. They do some of the food digestion, and they'll actually they pass nutrients around as they move. So if food was caught and gathered by a certain group of cells on one part of the sponge, some of the nutrients and energy is digested and carried by the amoebocyte and brought to another area of the sponge that maybe hasn't gotten a little bit of food in a while. And that way they actually share the nutrients. Again, this is just a loose colony of cells that are working together for each other's common good, and essentially that's what animals are. So the other thing that they do is they're the ones that create the spicules and the other support system, the collagens and the spongin that is used to create the skeletons of the system. The amoebocytes actually secrete those chemicals and make those structures. So essentially they're like the little builders and helpers that do everything with inside the cell. They don't do all of it, but they do help do a good bunch of stuff that's in there. So yay. So we have the pianocytes, the porocytes, the choanocytes, and the amoebocytes. So here's a picture of some of the different types of cells. So here's a choanocyte, the collared cell. Here is the cell, here is the collar. You can see that as the flagella beats, water is moved inside, has to go through the net. Things are caught and filtered out through the net and then digested by the cell or passed on to the amoebocytes for digestion. Here's a porocyte. Again, this is a cell that runs through and acts as a channel, not just a channel. But you can see here is the propofol, and it goes through and water would come out of the apical, and this would be the internal opening, the osculum, or the spongocyl, the cavity in the middle of the sponge. And then here is a actual slide picture of some of the different types of cells, and you can see where they're located. Like here's a bunch of the choanocytes all on the inside here. Here you have the mesohyl, the area between the outer layer of cells and the internal layer of cells. And you have some of the pianocytes acting as the skin on the outside, and the amoeba cells, which are passing things around and secreting the spicules and other spongin and stuff that's on the inside. So again, it's just a loose connection of cells working together for a common good, and that's the basis of an animal.